Welcome back, BioBuds. We are now in Unit 4, and we are going to be talking about topic 1, 2, 3, and 4, which is all about cell communication and signal transduction. So we have to understand, why do cells need to communicate with each other? Because it's very important for their function and survival. Well, think multicellular organisms are made up of many cells, and those cells cannot act as an individual organism by themselves. Therefore, they are going to have to communicate with one another in order to grow and to develop into all the many different parts of that organism that are working alongside one another. So cells are going to be communicating in 3 general ways. 1, direct contact, 2, local signaling, and 3, long-distance signaling. They're pretty self-explanatory, but let's dive into each one of those. So with direct contact, I'm talking about where there's a communication between cells that are literally in direct contact with each other, and they are communicating through cell junctions. So signaling substances and other materials are going to dissolve in the cytoplasm and can move from one cell to the next cell. Because these cells are touching one another and they have some connection port between the 2 cells to where that dissolved substance in the cytoplasm can freely pass. So if we're talking about animal cells, these are going to be gap junctions. So in this picture here on the left, our gap junction channel, notice that it goes directly from one cell to the next adjacent cell, and that these are almost like little almost transport portals, little tubes that are allowing the connection between the 2 cells. In plant cells, this is going to be our plasmodesmata that we talked about earlier in Unit 2. So plasmodesmata are holes in the cell wall of plant cells that allows for plants to exchange materials. An example of direct contact are going to be your immune cells. So these antigen-presenting cells are going to communicate to T cells through direct contact. So if you look at the picture, you have a T cell here that is directly in contact with your antigen-presenting cell. This is direct contact because they are touching one another and they are exchanging material via a gap junction. So think of it kind of like when jets are like refueling in the air. They have to have direct contact. They're not just like dumping fuel in the air above the airplane and just hoping that it goes in, right? There's a connector tube that they have to connect in order to transfer that liquid. So that's what we're talking about with direct contact. Local regulators, this is whenever a secreting cell, so a cell that is releasing things, will release those chemical messages, which are called local regulators and ligands. So this is an important word, and we will come back to this word later, but ligands that travel a short distance through the extracellular fluid. The chemical message will cause a response in a target cell. So we're going to have 2 options, and we are definitely going to need to know these words and be able to identify them. Paracrine signaling and synaptic signaling. I cannot talk. Synaptic signaling. So with these local regulators, one of our types is paracrine signaling. So this is whenever your cell that is doing the secreting, aka secretory cells, release local regulators, for example, a growth factor via exocytosis. So this is a vocabulary word that we have had before whenever we're talking about cell transport to an adjacent cell. So whenever we're looking at our picture, what do we notice? We notice, 1, that these cells are not in direct contact with each other. So that lets me know that it's not a gap junction. But they're close. All right, the next thing is that I have these molecules that seem to be going from one to another. I have this divot in the cell membrane, and here you've got vesicles, which indicate either exocytosis or endocytosis, but the fact that it's releasing things here lets me know it's exocytosis. So that is paracrine signaling. Synaptic signaling is happening in your nervous systems, okay? Our biggest example of this is going to be neurons, which are your nervous cells. They're going to be secreting neurotransmitters. So these are chemicals that are going to go from your neurons to other cells in order to signal a response. So once again, notice I am not in direct contact, but I'm close. So the cells are right beside of each other, but they're not directly touching each other. Once again, I have vesicles that are doing exocytosis here and releasing neurotransmitters and going to the target cell, the intended cell for that message. Then you have long distance signaling. So long distance signaling is typically going to be using hormones. In plants, an example of the use of hormones is going to be releasing hormones that travel through the vascular tissue, so xylem and phloem, the tubes that transport water and other nutrients throughout the plant, or through the air to reach target tissues. In animals, this is going to be dealt with endocrine signaling. So this is whenever you have specialized cells that release hormones into the circulatory system where they reach their target cells. So for example, your pituitary gland would be endocrine, and it's releasing hormones that are causing other things in your body to happen at different locations. So your pituitary gland is in your brain, and it is sending signals to target cells all over your body. Think about whenever you are going through puberty. It's not all about the brain, right? There's lots of other changes happening to your body that is being signaled in this situation. Another example of long-distance signaling is going to be insulin. So we know insulin's released by the pancreas, which is in your abdomen area, and the insulin's going to move into your bloodstream and travel throughout your body to bind to your target cells that need assistance with blood sugar regulations. So let's take a moment to quickly review these types of transport and signaling. So go ahead and pause this, get your answers, come back, and we'll see if you're right. So number one, what type of communication involves a cell secreting a substance to an adjacent target cell? Well, it's going to be paracrine signaling. So specifically, it's going to be local signaling. The fact that it's not involving neurotransmitters, but it's just a substance, kind of can help narrow it down to paracrine signaling. Number two, plant cells in direct contact with each other can diffuse substances through these structures to communicate. What are they? So I know that this is my direct contact because it tells me direct contact. And it specifies a plant cell, so I know that it's going to be plasmodesmata. So now that we've talked about our different types of signaling, let's talk about what happens whenever you actually receive that message. So whenever we're talking about cell-to-cell messages, we can divide that into 3 stages. The first one is reception. So think about like what it means to receive something. It means to get it. So this is whenever you have a ligand that binds to a receptor. So a ligand is represented by these. So it's just a chemical substance sent by one cell to another. So it's a very general term. There are many things that can be considered ligands. So for example, insulin would be an example of a ligand that's going to go and bind to another cell. Okay, so our ligand bonds to our receptor. So in this situation, this is my receptor, and I have this ligand that has bonded to it. And then my next step is going to be transduction. So that signal is going to be converted into a form that the inside of the cell is going to be able to utilize. So notice that my ligand actually isn't going into my cell here. It just attached to the receptor, and then the receptor then kind of changed and turned around and just shouted out, hey, we've got this here, this is what we're doing. The ligand doesn't actually come into the cell. And then we have response. So your cell is going to alter whatever it's doing in order to respond to that particular ligand and what that means. So looking at this one more time. So you have reception, you have a ligand that is binding to a receptor. You have transduction. That signal is being converted into these intermediate forms. A lot of times these are going to be like enzymes or other proteins. And then ultimately that's going to signal the cell to do something else. So that's going to signal the cell to stop breaking down sugar or start breaking down sugar, or it might be like a growth factor to where it's going to signal that the cell undergoes mitosis or stops undergoing mitosis. So we're going to look at each one of those in a little bit more depth. So with reception, we have, like we said, it's the detection and the receiving of a ligand by a receptor of the target cell. So my receptor is typically going to be a macromolecule that's going to bind to the signal molecule, which is the ligand. Most of the time these are proteins, but they can be carbohydrates and then even a few lipids as well. So all receptors are going to have an area that interacts with the ligand and an area that transmits a signal to another protein. And binding between a ligand and receptor is highly specific. So it's very similar to an enzyme situation where it's looking for a very particular substrate to bind to. Your receptors have a very specific ligand to bind to. So once again, if we look at this receptor protein here, we have an area where we are binding to a ligand and then an area that is going to be doing the trade. So once again, we have our receptor that is very specific, bonding to a specific ligand, and it has two separate portions. One portion is going to be where the ligand bonds, and the other portion is going to be where the transduction signal moves on. So when the ligand binds to the receptor activates via a conformational change. So remember, anytime you see conformational change, it just means some sort of shape change to where it's indicating that something else is going to happen. So this is going to allow the receptor to interact with other molecules to initiate that transduction signal. Receptors can be in the plasma membrane or intracellular. So it can either be within the plasma membrane like this one is, or it can be on the inside of the cell where the ligand actually has to enter the cell in order to have reception. So we have plasma membrane receptors, which these are the most common type of receptors involved in our signaling pathways. These are the ones that are going to be binding to our ligands, and specifically ligands that are polar and water soluble or large. Think back to our cell transport. These types of molecules do not easily pass through the cell membrane. Therefore, my receptor has to be on the outside of the cell because these ligands cannot move into the cell to have reception on their own. So some examples of these plasma membrane receptors are going to be G-protein coupled receptors, which is shortened to GPCRS. We'll go into more depth with those. And then also ligand-gated ion channels, which we will also go in depth on. Intracellular receptors are found inside the cytoplasm or inside the nucleus. They're going to bind to ligands that can pass through the plasma membrane. So if you remember back to our cell transport in Unit 2, these are going to be small hydrophobic nonpolar molecules. So things that can move through the plasma membrane to actually attach to my receptor inside of the cell. So some examples of things that would kind of follow this pattern are some steroid and thyroid hormones and gases like nitric oxide. So once again, here is a picture. This one's showing an intracellular receptor. So you have your signaling molecule that's actually moving into your plasma membrane, which is represented by that by this line here, this kind of grayish brown line. And within the cell itself, I have this receptor. So since the molecule can move through the plasma membrane, it can bind to a receptor on the inside of the cell. So this is intracellular reception. Stage 2, like we know, is transduction. So this is whenever you have an extracellular signal and it's being converted to an intracellular signal that can bring about a response. So this is going to require a sequence of changes known as a signal transduction pathway. So you're not going to have to know the names of like these specific changes of the molecules, but you just need to know that this is a signal transduction pathway. So think of it kind of like you're playing telephone. You can't move, but you can turn and like yell at somebody else, and you have to communicate from our classroom to the front office without actually walking in, without using a cell phone. So you have to just kind of like yell to the next person what you want to convey, and then they're going to turn and yell to the next person. And that is a signal transduction pathway. So the signal transduction pathway is going to regulate my protein activity, and it's going to do this via phosphorylation by the enzyme protein kinase. So we know phosphorylation means that it's gaining a phosphate, most likely from ATP. So this is going to be requiring energy. And there's this enzyme, protein kinase, that is doing this process. So this is going to relay my signal inside of the cell. Dephosphorylation, which is when I'm going to be removing a phosphate, happens by protein phosphatase. So this is going to shut off the pathway. So you might be asked which protein or which enzyme helps start the pathway and which enzyme helps stop the pathway. So for start, that's going to be a protein kinase via phosphorylation, which we know is adding a phosphate from ATP. Whenever we are asking what enzyme would stop a signaling pathway, that's going to be my protein phosphatase. So you should know that it has something to do with phosphates and proteins because it's protein phosphatase. So you're just going to have to remember that this one dephosphorylates, which means removing a phosphate. Okay, so by adding and removing our phosphates, that causes a conformational change, or for the protein to turn back to its original shape, which is what ultimately turns on or off the signal transduction pathway. So during transduction, the signal is amplified. This is done by secondary messengers or second messengers. So these are going to be small non-protein molecules and ions that help relay the messages and amplify the response. Okay, so our intracellular signaling molecules are our second messengers. An example of these are cyclic AMP, or just shortened to CAMP. You might see this quite a bit. This one is a common second messenger. So whenever we're talking about second messengers, we're talking about these guys here. So they are moving from where my receptor has had a conformational change, and they are going to inside of the cell in order to create our ultimate response that we were wanting. And that leads us to our ultimate response that we were wanting, our stage 3. So our response is our final molecule in the signaling pathway that's going to convert the signal to a response that alters my cellular process. So some examples of these: it can be a protein that alters membrane permeability. So maybe I needed to be able to let something in, but my membrane is situated where it will not let things in, but I need things to be able to let in. So it changes the ability of the cell to allow those molecules in that I needed in. It could be making an enzyme that's going to change a metabolic process. So that could be breaking down lactose. It could be breaking down blood sugar, things along that line. And it could also be a protein that turns genes on or off. So it could be where I need more of a particular protein, so I need to turn those genes on. Or I have too much of that protein, so I need to turn those genes off. So these are some pretty good examples that you should have an idea about, that these are common responses that you could encounter in your answers. So go ahead, pause this, look over these questions, and come up with your answers, and then check with me. So what are the 3 stages of cell signaling? You have reception, transduction, and response. Number 2, what is the actual signal being transduced in a signal transduction pathway? It's going to be your ligand, because your ligand is the molecule that bonds to the receptor to start off this whole process. And then how is the signal passed from outside to inside of the cell? Well, via transduction, because transduction, it's taking the signal that was relayed via the protein kinases and amplified by my secondary messengers. So my protein kinase caused my conformational change. It indicated that transduction needed to happen, that I had to turn on my signal transduction pathway. And then my secondary messengers are a part of that signal transduction pathway that brings that ultimate response to my cell. So our signal transduction pathways can actually influence how a cell responds to its environment. So like we said, they can result in changes in gene expression and therefore cell function. They can alter phenotypes or result in cell death. So a cell might get a signal that's saying like, hey bud, you're not quite functioning correctly, it's time to do apoptosis. And that could have been the signal that it was getting, to where it will complete apoptosis by breaking open its lysosomes and dissolving itself. It could, what it says by can alter phenotypes. Phenotypes is just the expression of your genes. So by turning a gene on or off, it can change the way that something appears. So for example, it's fall currently and the leaves are changing colors because the chlorophyll is being removed from the cells of the leaves as a pigment, and then the carotenoids are being left. So that's a change in phenotype that's caused by a signal transduction pathway that's ultimately due to lack of light or just the change in length of day. So sometimes you can have mutations in these receptor proteins, and what's going to happen, it's going to change the transduction of the signal. Because if I cannot bind to my receptor protein, then I might not be able to turn the signal on at all. So there's going to be 2 main categories of cell membrane receptors. We've already listed these off earlier, so now we're just going to go into a little bit more depth with these. The first one was our G protein coupled receptors, and our second one are ion channels. With our G protein coupled receptors, these are going to be our largest category of cell surface receptors, and they're very, very important in our sensory systems. So they're going to bind to something called a G protein, which is why it's called G protein coupled receptors. And this is going to bind to GTP. So it's very, very similar to ATP, just slightly different makeup. Doesn't have adenosine. So I have this GPCR, my receptor. I have an enzyme, and I have a G protein. And these are inactive until a ligand binds to my receptor. So let's label our picture here. This snake-looking guy here is my G protein coupled receptor. Okay, this is my G protein. These are coupled together, hence the name. Okay, so a ligand, step 1, is going to bind to my receptor. So then that's going to cause a conformational shape. That's going to cause the receptor to change shape. Notice the difference between here and here. The G protein activates and binds to my receptor. So that has happened here. where it was not attached here. And so that's going to activate my receptor and my G protein. And that is going to turn GDP into GTP. So I had phosphorylation going on there. So then I have the G protein that can bind to the enzyme. So notice I've got the enzyme being activated by the G protein, and that's going to amplify the signal and lead to a cellular response. So this is going to be my transduction pathway. So this is one type of receptor and how it causes its conformational change. So a ligand binds to the receptor, it changes the shape of the receptor, it allows this G protein to pair with it and to create this energy molecule GTP, and that helps with activating this enzyme here in order to start the signal transduction pathway. Our second type were ligand-gated ion channels. So these are going to be located in the plasma membrane, which makes sense because we're talking about like external ligands here. And these are very important to the nervous systems. They act as gates for ions. So that makes sense because this is ligand-gated ion channels. So ion channels, it's going to be allowing the movement of ions. And then the fact that it says ligand-gated, gated means that you can open or close. And then ligand is going to be your molecule that's going to bind. So when a ligand binds to the receptor, the gate opens or closes that allows diffusion of specific ions. So this is going to initiate events that can lead to a cellular response. So here's a picture of an example here where you have a channel that is closed. My gate is closed here. And whenever this particular neurotransmitter bonds to this ligand-gated ion channel, it opens and allows for ions to flow into the cell and out of the cell. And then once the ligand would release, it would signal the channel to close. All right, that wraps it up for topic 1, 2, 3, and 4 of Unit 4. Have a great day, guys.